Right, nucleic acid chemistry. In this session, we should be able to define what is nucleotide, define nitrogenous bases, list DNA and RNA nitrogenous bases, basically nitrogenous bases that are present in DNA and RNA, describe the general structure of purine and pyrimidines. So this is what I was telling you about. It is more, more into the molecular level of nucleotides. Describe the composition of nucleotides, how, what and all is present and what and all combine together to form a nucleotide. Mention the names of DNA and RNA nucleotide. It is different, it's not same. And how to name them, what is the nomenclature, what are all rules we have to follow while naming them. We have to also say, we have to describe the structure of RNA, basically. Okay. DNA is the DNA in structure and function of DNA. That will be your assignment topic, because you already studied it, right? You have finished the same in your first year. You have studied the same in phase 2. So I'm not going to take another class regarding the same. Okay, you have to study it yourself. And we have to classify RNA into 3 types based on their specific function. Overall, RNA has only one function, but that function is carried out by 3 different types of RNAs. We'll see how this function is subdivided and which RNA will be carrying out that function. Then you should be able to draw the diagram of tRNA and illustrate its structure, means label, neatly label that diagram, properly mark out the areas, label the different structures within the diagram, and finally you should be able to describe the structure of tRNA. So this is one of the important questions that you can expect in your examination. Structure of tRNA. Either you can get the nitrogenous base when describing nitrogenous bases, purines and pyrimidines. And the most commonly asked question is structure of DNA. You see your question paper, take your time to check the question papers. You can see the structure of DNA questions is being repeated many times. So when you are preparing for the examination, you have to make sure that you focus on these 3 topics and learn it thoroughly, so that it does not become a problem, because it is an easy scoring topic. Okay, so don't make it very hard. And finally, and structure and function of other 2 types of RNA, which you have to study. Examination point of view wise, this is not a very important topic, right? So let us start with nucleotides. So nucleotides are biochemical molecules. They are formed by 3 important components. Okay, they have a nitrogenous base. They have a pentose sugar, a carbohydrate, and a phosphate group. When these 3 things combine together, a nucleotide is formed. Okay, so nucleotides are found in both DNA and RNA. That is what our focus is. Along with that, these nucleotides are also having independent functions. Okay, as a nucleotide itself can carry out many functions throughout the body. They are very important, both structurally forming DNA and RNA, and functionally. Okay, the functions, independent functions within the body is also very significant. So nitrogenous base, pentose sugar together form a nucleoside. Okay, so what is a nucleoside? It is a combination of or composition of nitrogenous base and pentose sugar. When you add a phosphate to nucleoside, it becomes nucleotide. Okay, so please make sure you know the difference between nucleoside and nucleotide. Okay, so nucleoside is a combination of nitrogenous base and pentose sugar. Nucleotide is combination of a nucleoside and phosphate group. Okay, so these 3 components make up a nucleotide. Right. So So we'll see individual components of this nucleotide one by one, and then we'll go into the overall nucleotide. Right. So nitrogenous bases, these are defined as aromatic heterocyclic compounds found in both DNA and RNA. Okay. How do we define nitrogenous bases? Nitrogenous bases are aromatic heterocyclic compounds present in both DNA and RNA. There are of two types. Okay. So two types of nitrogenous bases are present. One is purine, other one is pyrimidine. Okay. So please make sure that you understand this two types. Why two types? These two types are important for us in the nucleic acids. Okay, purines and pyrimidines. And note down the spellings also. Very important. The spelling should be written properly. Okay, so let us have a recap of nitrogenous base. Nitrogenous bases are aromatic heterocyclic compounds found in both DNA and RNA. They are of two types, purine and pyrimidines. So let us see each one in detail. Again, purines are also heterocyclic aromatic compounds, and purines are also found in both DNA and RNA. So examples include adenine and guanine, which is found in both DNA and RNA. The chemical name of adenine and guanine is given below itself. And the chemical name of adenine is 6-aminopurine, while that of guanine is 2-amino-6-oxypurine. Now, this chemical name is very important when we are learning about the structure of these molecules. Right. So, general structure of purine. If you can see, there are two figures on the inside of your screen. So number one is the actual figure, and the second one is used to how to number the atoms. Okay, numbering the atom is very important. It gives us the structural explanation of functional group that is being present on the molecule. So you can see, as you can see, the general structure of purine is a two-ring structure. There are two rings. Okay, number one here and second one here. So it has two rings. It's a two-ring structure, and they are numbered in anti-clockwise direction. So when something moves from left to right, it is called as anti-clockwise direction. Okay. So here we start with here 1, 2, 3, 4, 5, 6, 7, 8, and 9. Okay. So numbering is done in anti-clockwise direction. So in this picture, you can see the chemical structure of adenine and guanine. As the name suggests, for the chemical name of adenine is 6-aminopurine. There is an amino group present at the sixth location or sixth position of the general structure. Okay. Similarly, if you look at guanine, the chemical name is 2-amino-6-oxypurine. So there are two functional groups present. One in the second location, that is the amino group. One in the sixth position, that is the oxy group. Okay. So that is how the structure is explained here. The general structure of purine, it is a two-ring structure. It is numbered in anti-clockwise direction. On the left side here, you can see the general structure of adenine, which has an amino group at sixth position, and guanine has two functional groups. One in the second position, that is amino group. One on the sixth position, that is the oxy group. Okay, you have seen purine. Let us see pyrimidine. Pyrimidine here is a single ring structure. Unlike purine, which had two rings, pyrimidine has only one ring in its structure. Just like purine, pyrimidine is also found in both DNA and RNA, but the nitrogenous bases, individual bases that are present in DNA and RNA, it differs when we look at the pyrimidine structure. Okay, so we have three pyrimidines that go into DNA and RNA. We have thymine, cytosine, and uracil. Okay, so thymine, the chemical name is 2,4-dioxy-5-methyl-pyrimidine. For cytosine, 2-oxy, 4-amino-pyrimidine, and uracil, 2,4-dioxy. pyrimidine. If you see the difference between an uracil, the only difference is the presence of methyl group in thymine. Okay, so thymine has only the methyl group that is present, which is absent in uracil. Now, this is very important. That's why we have three pyrimidine bases that are present in DNA and RNA, while only two purine bases. Okay, so that difference happens. I will explain it when we come to that. So, right now, please understand pyrimidine is a two-ring structure found in both DNA and RNA. In DNA, the pyrimidine nitrogenous bases that are present include thymine and cytosine. In RNA, it is cytosine and uracil. You see the difference here. Thymine and uracil is structurally similar, only difference is presence of a methyl group on the fifth position in thymine, but uracil does not have the methyl group present. So continuing on to the general structure, if you see here, again it is a single ring structure, is numbered in clockwise direction. Unlike purine, which was numbered in anti-clockwise direction, pyrimidine is numbered in clockwise direction. So these are the structures of individual pyrimidines that go into DNA and RNA. So cytosine, you can see there is a oxy group in the second position and an amino group in the fourth position. Similarly, thymine and uracil is very similar. The difference is a methyl group present in the position number 5, while that is absent in uracil. Now, this is very important. Okay, why this difference? I want you to keep that in mind. There is a difference between uracil and thymine, and the difference is presence of a methyl group. This helps us to understand why in RNA uracil and thymine cytosine is present. In DNA, thymine and cytosine is present. So that change in RNA, always thymine is getting replaced by uracil. Please understand this statement properly. It's always in RNA, thymine gets replaced by uracil. It cannot be ulta. Okay, it cannot be thymine replaces uracil because RNA is synthesized from DNA. Is a fragment of DNA is used to produce RNA in the within the cells, within the nucleus. So it's always important to know uracil is the one that is going to replace thymine when RNA structure is being formed. Right. So how to name nucleotides is very important. So what is nucleotides? It's combination of heterogeneous bases, a carbohydrate molecule, pentose sugar, and a phosphate group. So naming the nucleotides is very important. So it has some rules. Number one rule: prefix or the word that is added in front of the letter, the words which are added in front of the main word is called as prefix. Then prefix is given or prefix is named based on the type of pentose sugar that is present in the nucleotide. Okay, so again, let us understand this. Let me repeat it again. Prefix is always given on the basis of pentose sugar that is present in the nucleotide molecule. So based on the type of nucleic acid we have, there are two types of pentose that are involved in forming nucleotides. Number one is ribose sugar. Another one is deoxyribose sugar. Okay, so that's why we call it DNA deoxynucleic acid and RNA ribonucleic acid. So these names come based on the pentose sugar. We have ribose and deoxyribose. So in case the nucleotide is made up of deoxyribose sugar, then a prefix of D or D is given. Okay, so you have to add D in front of the nucleotide when the pentose sugar is deoxyribose. If the pentose sugar is ribose, there is no prefix should be added. So that is rule number one. Prefix for deoxyribose sugar is D. There is no prefix for ribose sugar. The second rule is adding the suffix. Okay, suffix is one that is added after the word. So suffix is added based on the type of nitrogenous base. So we have two types of nitrogenous bases, purines and pyrimidines. When purines are present within the nucleotide, the suffix of sine is added. When the nucleotide is consisting of a pyrimidine nitrogenous base, then a suffix of dine is to be added. So let me again explain the two rules. A prefix of De or D is added when the pentose sugar that is present in the nucleotide is deoxyribose. If the pentose sugar is ribose, then no prefix is to be added. Rule number two, suffix is added and the suffix is named on the basis of nitrogenous base that is present within the nucleotide molecule. If the nitrogenous basis is purine, then a suffix of sine is to be added. If the nitrogenous base is pyrimidine, the suffix of dine is added. If there is and it is named on number of phosphate groups that are present. If only one phosphate moiety is present, then it is called as mononucleotide. If two, it is di. If three, it is tri. Okay. So these three rules are to be used when naming the nucleotides. So naming the nucleotide is important because based on the type of looking at the nucleotide, we can estimate whether it will go and form DNA or RNA. Okay. So it is very important. So here are the names of nucleotides. On the left, you can see under the base, there are 4 nucleotides given, and this is for ribonucleoside. Okay, that means the top area, this area of within the table is meant for nucleotides that contain ribose sugar. Okay, as the rule suggests, we will name them. Adenine is a purine. The sugar here is ribose sugar. So there is no prefix because it is ribose sugar. And if suffix of sine is added, so adenine becomes adenosine. Okay, adenosine. And the nucleotide name, only one phosphate is present. That phosphate is binding at the 5th position. So adenine, when the base is adenine, when the sugar is ribose sugar, the name of ribonucleotide will be adenosine monophosphate. Okay, let me repeat again. Within a ribonucleotide, there is no suffix added, sorry, prefix is added, no prefix is added because the sugar is ribose. Suffix is added as sine because the nitrogenous base is purine. So, and it has only one phosphate group. So, the name of that ribonucleotide will be adenosine monophosphate. Similarly, guanine becomes guanosine monophosphate. Cytosine becomes cytidine because dine is the suffix added for pyrimidine bases. Okay, so it becomes cytidine monophosphate, and uracil becomes uridine monophosphate. And if you can see here, the abbreviation remains very simple. AMP standing for adenosine monophosphate, GMP standing for guanosine monophosphate, cytidine monophosphate, and uridine monophosphate. So these are the names of ribonucleotides. That means these nucleotides will go and form RNA. If you see at the bottom, the sugar changes. Okay, the sugar here is deoxyribose. So whenever there is a deoxyribonucleoside present, okay, the sugar is deoxyribose, then we have to add a prefix. Okay, prefix is to be added. So for a purine base of adenine, the sugar is deoxyribose. So the name of ribonucleotide becomes deoxyadenosine monophosphate or D-adenosine monophosphate, which is abbreviated. The short form is small d, then capital AMP. Similarly, guanine forms deoxyguanosine monophosphate sign because the suffix is given based on the purine base, that is sign. It becomes DGMP. Cytosine is a pyrimidine base, so the name becomes deoxycytidine 5 times monophosphate. It is called monophosphate. That much is enough. Deoxycytidine monophosphate is enough, abbreviated as DCMP. Thymine becomes deoxythymidine monophosphate. Continuing on into ribonucleic acid. So what we'll do is we'll have an summary of what we have learned, and we'll stop it in today's class. We'll continue it in next class. Okay. So just a brief summary what we have studied. We started with nucleotides. So these are biomolecules, biochemical molecules formed by composition of three things: purine, nitrogenous bases, pentose sugar, and phosphate group. They go and form DNA and RNA, which are the structural components of DNA and RNA. Along with that, they have independent functions. At the end, we had seen each nucleotide, both ribonucleotide and deoxyribonucleotide, these have independent functions in the body. So nitrogenous base and pentose sugar combine together to form a nucleoside, while phosphate group is added to a nucleoside to form nucleotide. Right. So next was nitrogenous bases. Both purine and pyrimidine are found in DNA and RNA. They are of two types, as we have seen. So purine bases, heterocyclic aromatic compounds found in both DNA and RNA. Both adenine and guanine are seen in both DNA and RNA. There is no change. Okay, so general structure, it is a two-ring structure. It is numbered in clockwise direction. Adenine is named 6-amino purine because presence of amino group at the 6th position. Guanine is called 2-amino 6-oxypurine. There is a amino group at 2nd position and oxy group at 6th position on the structure. Pyrimidine again, unlike purine, it's a single ring structure found in both DNA and RNA. Three examples: thymine, cytosine, and uracil. Thymine and cytosine is present in DNA. Uracil and cytosine is present in RNA. Uracil is present because thymine cannot bind to ribose sugar. It can only bind to deoxyribose sugar because the presence of methyl group within the thymine structure prevents the combination with ribose sugar. So general structure again, a single ring structure numbered in clockwise direction. You can see the structures of cytosine, thymine, and uracil. So naming the nucleotides, prefix is given based on the type of pentose sugar it is present. If the sugar is deoxyribose, deoxy D or D is given as prefix. If the sugar is ribose, then there is no prefix is added. Suffix is added on the basis of nitrogenous base. If there is a purine present, then the suffix of sine is added. If it is a pyrimidine, then diene is added. And based on the number of phosphate groups present within the nucleotide molecule, we either call it mononucleotide if one phosphate is present, dinucleotide if two phosphate moiety is present, and trinucleotide if three are present. So here you can see different nitrogenous spaces on the left column. In the middle, the nucleoside name, and the third column is the nucleotide, and at the end the abbreviation given for this. So using the rules, we have to name.